In this video, we will discuss about this characterization of this inducible cap system or iCAP system from the findings that they have find out in the previous experiment that we have discussed in our previous video. That is when the phage which is sensitive to the heparosan residues on the bacteria, or it can act on the bacteria where heparosans residues are present. So, and what researchers have found out, it's the KIFC that is mainly responsible for this heparosans residue on this cap. So if they inhibit this KIFC, then or they control the regulations of this KIFC, then they can control the heparosan levels on the cap and thereby control the viability of this bacteria. So how did they make this system? Is they make this KIFC genes controlled by this LAC1 promoter. So in this case, what will happen? And this LAC1 promoter is controlled by this small molecule inhibitor, that is IPTG. So when there is no IPTG, this LAC1 promoter will not be translated, and this KIFC genes will be translated, and it will form this heparosan residue on the membranes of the bacteria, and that will leads to the detection by phage, and it will reduce the viability of the bacteria. And when this IPTG is introduced, then it will lead to the formation of this LAC1. It will lead to the translation of this LAC1 promoter, and that will prevent the KIFC gene translations, and there will be no heparosans, and it will increase the viabilities or number of bacteria in the system. And they validated their system using the SDH phage experiment, where they quantify the cap formations, and they are able to see that with increase in the concentration of IPTG, they are able to see the increase in the amount of relative density of the cap. And when they did the time dependent assay and check the CAP dynamics, that is when they use co-culture, when they co-culture phage and this IPTG, because the IPTG will prevent the formations of heparosans, they are able to see an increase in the relative density of this cap. But when the IPTG is been removed, then they are able to see a decrease in the relative density, because now the bacteria will be able to form able to secrete this heparosan residue, or it will be able to translate this KIFC that will form heparosans on the membrane of the bacteria, and that leads to detection by the phage, and it leads to the death of the bacteria. And they also wanted to check whether this also has an impact, this IPTG also has an impact on the phage thickness and the polysaccharide length or not. So when there is no IPTG, they are able to see the thickness is around 44 nanometers, but when this IPTG is introduced to this, let us first discuss this 1000 nanomolars, they are able to see increase in the thickness to around 81.5 nanometers. It is 44.1 to 81.5 nanometers. And when they use intermediate concentration of IPTG, they are able to see the bimodal distributions, that is around 81.9, almost same. So this could indicates that IPTG does not affect the polysaccharide length, but it will affect the amount of the cap that are formed in the system. This is what they have concluded from their thickness analysis. So I hope you understood how they validate their system, or how they make the inducible encapsulated system, where they control the heparosan levels through this by regulating this KIFC gene through the LAC1 promoter, and which is controlled by the small molecules inhibitor, it is IPTG. So when there is no IPTG, this LAC1 will not be able to translate, and there is no problem in the KIFC translations. But when this IPTG is there in the system, then this LAC1 promoter will be translate, and it will inhibit the KIFC genes translations, and thereby these heparosans will not be able to secrete, and this will make them, and this will increase the viability of this bacteria against the phage. I hope you understood their system and how they validate their system. If you need to understand more about their system, that is inducible cap, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.